Looks like we're doing chapter 15, microbiology, and this is adaptive immunity, cell mediated immunity. And let's scoping out the chapter, you definitely need to know what an antigen is. So, antigens are red flags of foreign invaders. So, infectious agents carry around molecules that are unique to them. They can provoke the immune cells to go into attack mode. These antigens can be complex, whole microbes, or parts of microbes, such as the toxins they release. We're going to talk about lymphocytes. Lymphocytes are the skilled troops of immunity. So during development, millions of lymphocytes in the bone marrow each become programmed to go after a specific infectious agent and are primed for future battles with these microbes. Eventually, they are deployed into lymphatic tissues throughout the body to be ready for attack. And in getting prepared for battle, the captured microbes are taken in by macrophages, where they are imprisoned and reduced to their most antigenic parts, broken up in the antigen. And so the macrophages then display the antigens and make them available to the lymphocytes. This brings about an activating process which prepares the T and B lymphocytes for their extended roles. We'll go more in depth with that. Then we're going to talk about T cells. There's T helper cells and T killer cells are cytotoxic T cells. So once they have been properly stimulated, the T cells go down one of two different pathways, leading either to helpers, T helper cells, that assist other cells get ready to fight infections, or to killers that are involved in cell to cell combat. These cells bring smart bombs that can take out foreign cells with great precision. Then we have B cells and antibodies. This is a protein defense. So stimulated B cells go into an active growth mode, making many clones of themselves that will produce their key defense molecules, which is the antibody. These weapons are released in circulation to surround and hold microbes hostage so that they can be dealt with and destroyed. Then we're going to talk about vaccines and other therapies to mimic nature. So, one significant advantage of having an organized and well equipped strike team is that it can be trained for future conflicts. So, if an antigen is carefully selected and processed, it can be used to induce immunities before an infection has even occurred. So let's look at the third line of defense, adaptive or acquired immunity. So product of B and T lymphocytes dual system. Okay, so this relates to our immunocompetence. Immunocompetence is the ability of the body to interact with a wide spectrum of foreign substances. So we're going to look at antigens. Antigens are molecules that stimulate response by T and B cells. Two features that characterize immunity: specificity. So, with specificity, antibodies are produced and they function only against the antigens that they were produced in response to. That's why they're very specific. So, an antibody is very specific for an antigen that it's produced for. Then we have memory. So, lymphocytes are programmed to recall their first encounter with an antigen and respond rapidly to subsequent encounters. So here's an overview of the specific immune responses. So separate but related activities of the specific immune response. So first, you have development and differentiation of the immune system, the lymphocytes and antigen processing, the cooperation between lymphocytes during antigen presentation, and we'll go over all of these in more detail. Then we talk about B lymphocytes and the production and action of antibodies, and then T lymphocyte responses. Okay, this is the lymphocyte. You have a lymphocyte, and there'll be We'll have other slides that kind of describe this. But this lymphocyte is made in the bone marrow. So the lymphocyte is made in the bone marrow. Some of them will go to the thymus. That's the T cell line. They will produce, and they mature in the thymus, and now you'll have a T lymphocyte. And then it goes to the lymph node or other lymphoid organs. Okay, so that's T lymphocytes. Then the bone marrow produces immature lymphocytes, and if it stays in the bone marrow, goes to a different part of the bone marrow, and it matures and becomes a B lymphocyte. And that B lymphocyte then goes to a lymph node or lymphoid tissue as well. So, development of the lymphocyte system. So, lymphocytes arise from the same stem cells but differentiate into two distinct cell types early on. So, T cells mature in the thymus, B cells mature in bone marrow. So, once they're released, the mature cells settle in lymphoid organs and serve as constant attack force for infectious agents. And we went through this figure in class. This here is a macrophage or phagocyte. It engulfs or takes in things. And so in this case, if there's an antigen, which is a foreign invader, and this macrophage, which this phagocyte is called a dendritic cell, and they're the most prevalent. This identifies something that's not like itself, and so it will engulf it or take it in. So this one is one that we talked about in chapter 14 that is non-specific defense. So non-specific means that it's not specifically going after this antigen. It just knows that it's something that's not like self. It's not self. And so it will engulf anything that's not self. So the antigen's engulfed, the dendritic cell breaks it down, takes little pieces of it, puts pieces of it, and presents them outside of the dendritic cell. So it presents it outside. Now, this T lymphocyte has a receptor to a specific antigen. And it's specific to this antigen here, and that's why it comes and it recognizes that this antigen is the one that it is specific for. That's the T helper cell. At the same time that this is doing this, then you have B lymphocytes, and they have receptors on their surface as well to a specific antigen. And in this case, it's this specific antigen. And so it's being exposed to this antigen as well. And it's saying, okay, this is an antigen that we need to go to war against. Oops. The antigen that we need to go to war against, but it's not activated. It's waiting to make sure that they get confirmation. So the T cell, if its antigen receptor comes in contact with the antigen that's on this dendritic cell that's being presented, then it will go and it will confirm with the B cell that the antigen that it's sensitive to, that it's an antigen receptor that it recognized, that it is definitely a non-self antigen, and this B cell gets activated. Once the B cell or B lymphocyte becomes activated, it becomes an activated B cell, and it can go one of two ways. It can make more of itself and keep a memory of that antigen. And so it's a memory B cell, and that's where you can get memories for years once you've been exposed to something. and also it can produce plasma cells. Plasma cells will just produce a lot more of these antibodies, this antigen receptor, just like these antigen receptors that are on the surface here. These are actually antibodies or immunoglobulins. So it can produce plasma cells that will make a lot more immunoglobulin, and they'll release immunoglobulin into the blood, into the interstitial tissue, into the lymph. It just sends antibodies everywhere. And these antibodies will fight this specific antigen. So it's very specific. Okay. So that's when the T helper cell activates the B cell. Then once the T helper cell communicates with the B cell that has that same antigen on its antigen receptor, and they agree that that is a antigen that is not a self antigen, then this T helper cell becomes activated as well. And with the activated T helper cell, activated T cell, it will make memory T cells. So if they ever come across this again, they'll already be aware of what needs to be done to neutralize it. So that's becomes memory T cells, and also releases chemicals to bring other immune responses to the area. And then it goes down and it will differentiate into produce more T helper cells, or you'll have a cytotoxic T cell. That's the T cell that can kill the invader directly. So once it's activated, it can produce memory T cells, or it can go down and produce more helper T cells or cytotoxic T cells. Okay, so you have contact with the antigens and then presentation by antigen presenting cells. So foreign cells bear molecules or antigens that are recognized and engulfed by antigen presenting cells, such as the dendritic cell. So in that previous slide, it was a dendritic cell, but it was an antigen presenting cell, which means it takes the antigen in, engulfs it, chops it up, and then takes a piece of it and presents it on its surface. So for most responses, the T helper cell first receive the processed antigen from the antigen presenting cell, and then they go on to activate the B cells and other T cells. So then you have activation of the T cell. So then the T cell responses. An activated T cell forms memory cells and differentiate into helper T cells or cytotoxic T cells. So T cell immunity is termed cell mediated because the whole T cell acts directly to destroy microbes rather than by secreting molecules into the body fluid. So the T cell itself the cytotoxic T cells will actually kill the microbes directly. Then you look at B cell responses. So a B cell activated by a T helper cell undergoes a spurt of cell division. So it produces memory cells that provide a rapid recall of the antigen, and then it provides plasma cells that secrete proteins called antibodies. So all of that is what we described here. So activated B cell will make memory B cells, or will make plasma cells that produce a lot of antibodies. And once again, we talked about an activated T cell. It will produce more memory T cells, and then it will also produce helper T cells or cytotoxic T cells. Cytotoxic T cells directly kill the microbe. Then we have what we call humoral immunity. So antibodies circulate in fluids, the blood, extracellular fluid, and lymph, providing humoral immunity. Know where the antibodies actually circulate in all these different areas. So the antibodies react specifically with the antigen and mark it for an enhanced response. We'll go through that as well. So development of the immune response system. So we have cell receptors or markers confer specificity and identity of a cell. So major function receptors, they perceive and attach to non-self or foreign molecules, promote the recognition of self molecules, and receive and transmit chemical messages among other cells in the system. They also aid in cellular development. So the major histocompatibility complex. So set of surface proteins, which are receptors, they're essential for acquired immune system and the recognition of self and the rejection of foreign molecules. I'm not going to ask you about major histocompatibility complex. We can read through it though. So it talks about type 1, type 2, or class 1, class 2. Like I said, I'm not going to actually talk about that. So let's look at lymphocyte receptors. Remember, you have a B lymphocyte, T lymphocyte. So the role that lymphocytes play in surveillance and recognition is a function of their receptors. So the B cell receptors, they bind free antigens. So if antigen is floating around, the B cell receptor just binds the free antigens. The T cell receptors, they bind processed antigens together with the MHC molecules on the cells that present antigens to them. And so in this case, we were looking at, this is where what it's talking about in terms of it has antigen receptors on the B cell, and it interacts with free antigens directly. The T cell has an antigen receptor, but it only responds to this antigen that's being presented by another organisms, another cell. Okay. All right. So that's where B cell receptors bind to free antigens. T cell receptors bind to process antigens. Okay. Antigens are molecules, so their chemical structures can vary over a very wide range. So it's potentially exhibiting billions of uniquely different structures and shapes in that antigen receptor. Okay. Sources of antigens include microorganisms, chemical compounds in the environment. So your immune system sets up, even if it hasn't been exposed to them, has receptors for billions of potential invaders. Actually, we went over clonal selection theory in lecture. I'm not going to ask a question on that though in the quiz. So development of lymphocytes. In the bone marrow, lymphocytic stem cells differentiate into either T or B cells. B cells stay in the bone marrow, while T cells migrate to the thymus. Then both T and B cells migrate to secondary lymphoid tissue. Secondary lymphoid tissues will constantly be resupplied with B and T cells. So this is just another physical diagram that shows lymphocytes are made in the bone marrow. So this is lymphocytes. This is immature lymphocytes. If this line goes and stays in the bone marrow, just in stromal cells in the bone marrow, then these lymphocytes will mature into B cells, which has these immunoglobulins on the receptors of this B cell. Then from there, this B cell goes to lymphoid tissue. Well, here's another uh, lymphocyte that if it came over this way, didn't stay in the bone, but went to the thymus. Then it would develop these uh, antigen receptors on its surface, and it is a T helper cell. And then it's released into the lymphatic tissue as well, lymphoid tissue. So lymphocyte specificity is genetically pre-programmed even before an antigen has ever entered the system. So the lymphocytes are each cloned. So each genetically different type of lymphocyte expresses a single specificity. So what they're saying here is this one of them clones itself billions of times. And then each one of these develops a receptor that's independent of all the others. And so this receptor is the antigen receptor. If they ever came across a organism that had that receptor, then it would activate this specific uh, lymphocyte. So when they're developing, billions of lymphocytes are made, each of them having a different receptor, antigen receptor on it. Each one of these is a little bit different. So when you produce a whole lot of them, there will be some that actually don't recognize self as being self. And so they actually have antigen receptors that if they're part of the antigens that make up our human body. So if there's some lymphocytes that do not recognize ourself as self, then they are eliminated. And so then what you end up having is you end up with a repertoire of lymphocyte clones. So they're exactly alike, except each of them has a unique receptor, antigen receptor. That's the only difference with them. I'm not going to ask about antigen independent. Okay. All right. So when you end up having a set of lymphocytes that have these receptors for antigen, so if you have entrance of an antigen and it actually is recognized by this lymphocyte, then it will activate and it will start reproducing itself and it'll start making more antibodies. So that's basically what this is saying. We'll talk about that. Now let's talk about the specific B cell receptors. Let's talk about immunoglobulin. Immunoglobulin is also called antibodies. And let's see, they have antigen binding sites. I'm not going to have you get into this real specific, but know that immunoglobulins can actually attach to two different antigens. And this is where it has the antigen binding site. Okay, then we talk about development of immunoglobulins. So you have undifferentiated lymphocytes, and then they differentiate to be receptive to multiple different antigens. But once one keys in on one, then that one's just specifically for that antigen. But you have billions. Okay. Let's see. Don't have to worry about the V and D segments. Don't have to worry about technical pictures, heavy light chain. Just know that their immunoglobulins are considered antibodies. You don't have to worry about that. Then so we have T cell maturation. Its maturation is directed by the thymus gland and its hormones. So you have different classes of T cell receptors, and they're termed CD receptors or clusters of differentiation. So you have CD4 on T helper cells. That's the name of the T cell receptor, CD4. Then you have called CD8 on the T cytotoxic cells. So mature T cells migrate to lymphoid organs. So B cell maturation is directed by bone marrow sites. So B cells will come in contact with antigens throughout their life. And so they'll have immunoglobulin as surface receptors for antigens. And so you contrast the properties of T and B cells. You won't really have to know this table. So antigen is a substance that elicits immune response in specific lymphocytes. So if you have antigenicity, that's the property of behaving as an antigen. So it's all related to foreignness, which means if it's not self, its size, shape, and how accessible it is. So antigens have many antigenic determinants. These are called epitopes. They're a small molecular group recognized by lymphocytes. Just know that they're antigens. And so this is saying that the site that's the antigen receptor is actually receiving the epitope, which is the site that this antigen receiver fits in. So you have foreign cells that are just completely different, and then also large molecules greater than 10,000 molecular weight are the most antigenic. They will cause the most response. So complex molecules with several epitopes, several areas that the immunoglobulin will respond to, make good immunogens. That means it will elicit a response. Poor immunogens don't elicit much immune response. There are some small, simple molecules that aren't attached to a carrier molecule. And then some simple, large molecules as well. So if you just have a repetitive chain of monomers, the same thing over and over, those aren't very, don't cause much reaction. So yeah, the epitopes, these are the epitopes, and they fit onto the active site of the immunoglobulin. And then those are the ones that actually will attach to. You don't have to know those as specific. Haptens are small foreign molecules that consist only of a determinate group, and they're too small by themselves to elicit an immune response. Okay. So Talk about superantigens. These are potent T cell stimulators. And that's like toxic shock syndrome has a massive release of cytokines leading to cell death. You have allergens, which are antigens that evoke allergic reactions, typically in things that most people do not have an allergic reaction to. And then you have autoantigens. This is molecules on self tissues for which tolerance is inadequate. All right, cooperation immune reactions to antigen. So the basis for most immune responses is the encounter between antigens and white blood cells. The lymph nodes and spleen concentrate the antigens and circulate them so they will come in contact with lymphocytes. Remember we said the B lymphocyte and the T lymphocyte, they actually go to the lymphoid tissue and that's where they're stored. Now, the way the lymphatic organs are situated, the lymph flows through them and they actually filter. So anything that is in the lymph is actually going to show up in the lymph nodes and spleen. That's how they're able to concentrate the antigens. If there is an antigen in that area, it'll get concentrated. And then the B cell and the T cells, T lymphocyte, B lymphocyte are already there. And then so the T cell dependent antigens must be processed by phagocytes called antigen presenting cells. This is this is the same thing that we're talking about with that figure. So you have three cell types that can serve as antigen presenting cells. You can have macrophages, dendritic cells. The example we were using was the dendritic cell. And then B cells can act as antigen presenting cells as well. So antigen presenting cells modify the antigen. Then the antigen is moved to the APC surface and bound to an MHC2 receptor. Won't tell you you won't have to go through the interleukins. So this is another schematic that's the same thing. These are foreign microbes or antigens taken in by a dendritic cell, and the dendritic cell chops it up, takes a piece of it, and puts it onto the surface. So this is an antigen receptor, and it puts this on its own surface, this dendritic cell. And so then when the T cell that has that same antigen communicates with this antigen that's on the macrophage, then it goes into it goes and it connects to a B cell, and the B cell has a receptor on with that to that same antigen, and then if the T cell and B cell collaborate, that this antigen that they both are sensing is a foreign cell. Then it goes into those things I talked about where the B cell um, divides into a memory B cell or divides into a plasma cell. It releases immunoglobulins. Okay. I want to ask you specifically about APCs, MHC. So let's just go through here. Okay, cell-mediated immunity requires the direct involvement of T lymphocytes in the immune response. So the T cells secrete cytokines that act directly on other cells. So before a T cell gets activated, the antigen must be presented on the APC antigen presenting cell, which is that dendrite. Once activated, the T cell transforms in preparation for mitotic division to become a subset of effector memory cell. So differentiation will be determined by the antigen presenting cell interleukin secretion. You don't have to know these CD4, CD8, things like that. Interleukin, you don't have to know those. The T helper cells are the most prevalent type of T cell in the blood and lymphoid, regulate immune reaction to antigens, including other T cells and B cells. Also involved in activating macrophages and increasing phagocytosis. And so differentiation of T helper 1 or T helper 2 depends on what set of cytokines is released from the antigen presenting cell. So cytotoxic T cells destroy foreign or abnormal cells by secreting perforins and granzymes. Natural killer cells, they lack specificity. They circulate through the spleen, bloods, and drugs and lungs. And so T cell activation and differentiation. This is the dendrite. This is the T cell. It has on its surface, it has a receptor for the antigen. And so when the dendrite or the macrophage eats the antigen and breaks it up and put on the surface the antigen, then the T cell that has that antigen receptor, it goes and then it starts this whole process where it can go into memory T cell, activated T cell, and so okay, and this is where it does the same thing up here where this is the T cell and T helper cell, then it activates the B cell. All right, natural killer cells. Natural killer cells, they release perforins. It actually eats up the cell membrane of a foreign body. So natural killer cell is here. This is the foreign cell down here, and it releases perforins, which actually penetrate the cell, the invading organism. And so you have reactions. Reactions have drastic consequences sometimes. You can have a super antigen. They're found primarily in bacteria and viruses, form of virulence factor. And so it provokes an overwhelming immune response by large numbers of T cells, regardless of specificity. It says, just come to our area. So when you release massive amounts of cytokines, you'll get blood vessel damage, you get toxic shock, multi-organ failure. So then we've talked about the B cell responses, B cell activation, and antibody production. And divisions give rise to plasma cells that secrete antibodies and memory cells that can react to the same antigen later. We'll go through that when we see it. There we go. So this is that same figure, or close to it, saying that you have an antigen that gets presented on an antigen presenting cell. So when we looked at the dendritic cell, it actually puts on its surface a little piece of that antigen it engulfed. When this helper T cell recognizes that antigen, then it will activate the B cell. And then the B cell will go on to produce more B cells that are like itself. And it will differentiate into plasma cells will release antibodies. I'm not going to go in depth with clonal selection and antigen binding. This is what we were talking about, and actually, this just kind of describes this next. Let's see. Actually, it describes this previous slide here. So it just says clonal selection and antigen binding. So the B cells can independently recognize microbes in their foreign antigens and combine them with their Ig receptors. This is how the initial selection of antigen-specific B cell clone occurs. So it's just saying that this B cell, it can come in contact with this antigen on its own. That's basically all that was saying. And then it can get activated by the T cell. So the B cell activation, the T cell gives off additional signals and activates that B cell. Then you have the B cell will create a memory B cell, or it will go on to create plasma cells, and it will produce immunoglobulins. Then you have the plasma cells, which produce the immunoglobulins, or antibodies. And so the immunoglobulins, they have antigen binding fragments, and then they have a crystallizable fragment. So here's the active site where it can respond to a antigen. So that whole thing, this whole thing is an antibody immunoglobulin. We don't have to go into specifics on its site. Okay, here's some principal activities of antibodies. With antibodies, when they're produced, they unite with, immobilize, call attention to, or neutralize the antigen for which it was formed. Now, first, it's called opsonization, process of coating microorganisms or other particles with specific antibodies so they are more readily recognized by phagocytes. It's carried out by antibodies called opsonins. So that's opsonization. Neutralization. Antibodies fill the surface receptor on a virus or the active site on a microbial enzyme to prevent it from attaching. And we have agglutination, antibody aggregation, cross-linking cells or particles into larger clumps. Then complement fixation, activation of the classical complement pathways can result in the specific rupturing of cells in some viruses. Then you have precipitation. You have aggregate of particulate antigen. Here's different things here. The immunoglobulin or antibody can tag this cell. And then you have opsonization. Once these are all tagged, then it's easier for them to be engulfed by macrophage. You have neutralization. That's where all these antibodies will attach to foreigner, like in this case, this virus. And so this virus cannot infect the cell. Then you have agglutination. The antibodies hold to one, hold to the other, and they all stick together. Yeah, complement reactions. And then this is the antibodies come in and they have substances, they draw substances that can actually breach the cell membrane. We have precipitation. That's where the antibodies, if they have antigens and antibodies together, then they all come together and it creates a clump. Don't worry about the FC regions, antibodies, but I do want you to know the functional classes of immunoglobulins. So these are immunoglobulins, or antibodies, IgG, IgA, IgM, IgD, IgE. And know that the IgG is most prevalent. The IgA uh, is found in the mucus and serous secretions. IgM is the first immunoglobulin to be uh, activated, synthesized once a antigen is found out. And let's see, IgE, know that it's artistic response. Okay, all right, and also let's see, the IgG is the one that is able to cross the placenta. So it can cross placenta and possibly cause problems with the developing fetus. Okay, the antibodies in serum. So anti-serum are serum containing specific antibodies. You don't have to know the electrophoresis and all that. So regardless of the site where antibodies are first secreted, large quantity eventually ends up in the blood. And this is monitoring, this is if you've never been exposed before, IgM is the first in the scene. If you expose to an antigen, IgM is first in the scene, then you have IgG. And this is secondary response. If it's second exposure to the antigen, is almost immediately the IgG responds harder than the first time and sustained. This is why people can get immunized, and some immunizations last a lifetime. Other ones, the body has memory for, let's say, a couple of years. So won't worry about these responses. Now you can talk about monoclonal antibodies. They originate from a single clone and have a single specificity for antigen. And then, actually, we won't talk about monoclonal antibodies either. You don't have to worry about that. Okay, so the monoclonal drugs for treatment, you don't have to know those. This I do want you to know. Categories, immunity categories by mode of acquisition, how you actually got it. Active immunity results when a person is challenged with an antigen, stimulates production of antibodies, creates a memory. This takes time, but it's lasting. It's like you get exposed to something, you get sick, and your body fights it off and wins, and you keep an active immunity. That's active immunity. Passive immunity is preformed antibodies are donated to an individual, does not create memory, acts independently short term. So let's say if someone already fought off a disease and then takes some of their plasma and gives it to someone else, it'll fight off the disease in that person, but it does not give long-term immunity. This takes care of it right then. You have natural immunity. It occurs as a normal life experience. Then you have artificial immunity. It's acquired through medical procedures such as a vaccine. That's artificial immunity. So understand these: natural active immunity. So getting an infection after natural active immunity. After recovering from an infectious disease, a person may be actively resistant to reinfection. So it all depends on the disease. Then you can have natural passive immunity. That's where mother to child, the IgG antibodies from the maternal bloodstream can travel or be actively transported across the placenta to the fetus. So know that IgG can travel, and that's an example of natural passive immunity. So IgA antibodies from the mother's milk reacts against microbes entering the intestine of the baby. Then you have artificial active immunization. The vaccination, which is the microbial or antigenic stimulus, it triggers the immune system to produce antibodies and memory cells. That's artificial active immunization. And that's what, when you hear immunization, that's typically the one they're referring to. Then you have artificial passive immunization. And so that's immunotherapy. Patient at risk for acquiring a particular infection, administered a preparation that contains specific antibodies against that infectious agent. So that's basically saying you fight something off, you have antibodies against it, take your plasma, give that plasma to someone else, and the antibodies are in their body to fight off any disease. That's active passive immunization. Then you have acquired immunity. That's acquired immunity is natural immunity acquired through the normal life experiences. You have active immunity results when a person develops his own immune response to a microbe or infection. Passive immunity results when a person is given selected immune substances made by another individual. This baby is getting her mother's immunity through the IgE. You have artificial immunity produced purposely through medical procedures, also called immunization. So we went through these: passive immunity, artificial passive immunization. This is useful for immunocompromised patients who cannot be vaccinated. Acts immediately, protects the last 2 to 3 months. So that's artificial passive immunization. That's where, like, someone has the antibodies in their system, they beat it, and they take those antibodies and give it to someone prophylactically. It's almost like it's an immunization because it's almost like immunization because you just give it to them, have to give it to them again because they cannot make their own. So antisera and antitoxins of animal origin. Sera produced in horses are available for diphtheria, botulism, and spider snake bites. Okay, let's look at how vaccines. Most vaccines are prepared from killed whole cells or inactivated viruses. So most of them are made from, they just kill it, take pieces of it that will elicit immune response, and that's what they make the vaccine from. That is killed or inactivated. Then you have live attenuated. Attenuated means that it's live, but it's weakened, so it won't cause disease. So you have live attenuated cells or viruses. Then you have antigenic molecules derived from the bacterial cells or viruses. So they just take a piece of the antigen. Okay. Okay, here's a checklist of things, requirements that for an effective vaccine. Well, first, it should have a low level of adverse side effects and not cause serious harm. It should be able to protect against exposure to natural wild forms of the pathogen. Should stimulate both B cell response and T cell response. The B cell response is the antibody response, and the cell mediated response is T cell. It should have long term lasting effects, like producing the memory cells. It should work with minimal doses of boosters, and it should be inexpensive, have a relatively long shelf life, and be easy to administer. Different types of vaccines. So with this, it's saying if you use the whole cell vaccine, there's different types that people use, and they can kill it, or if the whole cell, they can kill it and inject the whole cell, make the vaccine from the whole cell, or they can just decrease the virulence of it and use the whole cell to make an antibody. And this just shows that you don't have to have the whole organism. You can just have pieces of it that would be the antigen. Yeah, antigens can be taken from cultures produced by genetic engineering or synthesized, made. And like, let's see, DNA vaccines. So you can have genetically engineered vaccines. DNA vaccines create recombination by inserting microbial DNA into a plasmid vector. So you just take a piece of the microbes' DNA, and that's the antigen. So the human cells pick up plasmid that's been incorporated in. They express that microbial DNA, express it as proteins. So it's causing the B and T cells to respond, that antigen, that protein. And so then they become synthesized and form memory cells. So there's experimental vaccines for Lyme, hepatitis C, herpes simplex, influenza, tuberculosis, and malaria. But typically it takes 10, 15 years to get a vaccine. So here's just a simple example of it. This bacteria or whatever died or whatever it did with it. But what it does is take a little piece of its DNA and you can put it into a plasmid with the DNA from the pathogen. And then you inject that plasmid with that DNA, goes into the human body, goes into the cell, and it starts duplicating, replicating this protein that came from the microorganism of interest. And so it produces this protein, and then the protein from the pathogen is expressed on the surface of this whole cell. Then all the defenses recognize it and build up immunity against it. And they have genetic engineered vaccines. I'm not going to have you go through those. You don't have to know those licensed vaccines. Uh, okay. So rights of administration and side effects of vaccines. Most administered by injection. Few of them are oral or nasal. Some vaccines require an adjuvant, which is actually just something, let's say some fluid to help it flow. So uh, you have to have more benefit than risk. And then look at some of the possible side effects. So whenever we know or suspect adverse effects been detected, then vaccines are altered or withdrawn. Depends on the adverse effects. Then a lot of people just hope for herd immunity. So some individuals will not harbor it. That's with herd immunity, enough people have immunity to it that it won't be harbored in them. It reduces the occurrence of that pathogen causing disease. That's called herd immunity. So it's less likely that the non-immunized person will encounter the pathogen. There's so many people around them that don't have it. And then they go through the recommended immunization schedule, which you don't have to know. Okay. All right, that is it. So what I'm going to do, I'm actually looking at my quiz. So in chapter 14, definitely know the components of the second line of defense. So, we talk about first line of defense and what that would actually include. And know the type of blood cells and what they target, like basophils, eosinophils, neutrophils, monocytes, what they target. And then know what a fever is and what's associated with. And then look at the different lymphoid organs, know what the lymphoid organs are and what they do. And know where lymphocytes are, where they mature. Definitely know the classic signs and symptoms of inflammation. Rubor, pallor, tumor, dolor. Know that. And so once again, which the blood cells that target different things. So know which one targets what. And what are the key phagocytic cells of the body? We have the neutrophils, the macrophages, the dendrites. Just know what the key phagocytic cells are. And then know genetic immunity, what that actually means, what that is. And then inflammatory response, know which line of defense it is. And then know the different antibodies, the names of how it affects microorganisms like neutralization, opsonization, complement fixation, agglutination, know all of those. And then know the classes of immunoglobulins. And so we have IgA, IgD, IgE, IgG, IgM, and know the major portion of each of them. There's a couple questions on that. And then understand the natural and passive immunity, the different types of immunity. Definitely know those. There's a couple questions on that. Know what clonal deletion is. And then and with vaccines, know the different types of vaccines that we have, like acellular vaccines, what they actually, acellular vaccines, like when they just take a antigenic part, not the whole cell. Okay, and so I think that pretty much is, and actually the antibodies, where they're located in the body. Okay, so I actually went through the quiz, gave you an idea on each of those parts. Study well. Definitely look at the um, Khan Academy and put the time in, and you'll do well on this quiz. I am looking forward to seeing you all Wednesday. All right. Have a good one.